Hello, ladies and gentlemen. Welcome to Mr. Sauceda's YouTube videos. Today we're going to be talking about how cells divide. We're not going to go over mitosis yet, though, because this is like a short day. So how exactly do cells divide? First things first, the nucleus of your cell contains all of your DNA. So the nucleus of any one of your cells has a complete set of genetic code to build any part of you and to build your entire body. So just keep that in mind. That's the reason why when they do a DNA test or something, they can just swab your cheek or something. All it takes is a cheek cell, and they can get your complete set of DNA that way. So, what do we need to know? Well, before mitosis, so before cell division, the DNA is inside of the nucleus. It's protected, right? The nuclear envelope is there protecting it. And so, not only that, but think about it. Here's a little plant cell image. I think it's the onion root tip that we kind of looked at before. But you can kind of see we have this nucleus. It's nice and protected. Every single cell in your body contains 9 feet worth of DNA if you were to stretch it out. It's a lot of DNA. How in the world, you're made up of like trillions of cells, right? Can every cell have 9 feet worth of stuff in it? Clearly, it has to be wrapped up somehow. So DNA is wrapped around proteins called histones, and they form a DNA protein mixture that we call chromatin. So it isn't just DNA, it's chromatin. DNA would be if you removed if you unwrapped everything, you'd have 9 feet worth of stuff. So we don't want that. We would also like the histones and stuff because it saves space. And so the histones are kind of like a like a spool for like thread or something, just kind of wrapping it around. So to conserve space, DNA will wrap itself around these proteins like yarn. And you don't need to know the details of this, but there are different parts to it. So you have this is the DNA all stretched out. Then you have what they call kind of the pearl necklace. And that's because you have little histones and the DNA is wrapped around itself like this. And then if you put enough of these together, you start to get, kind of these nice organized patterns. And then it'll start to get really windy. And then it'll start to compress itself into something that we would call a chromosome. So here's the thing though. Most of the time when your cell is not dividing, there are no chromosomes. It is literally just this kind of beaded necklace, wibbly, wobbly kind of stuff. It's chromatin. The only time that it organizes itself into chromosomes is when the cell is ready to divide. Why? Because think about it. You are about to create a complete genetic copy of DNA. You want to make sure you get it right. And your cell definitely wants to make sure it doesn't make a mistake. Because remember, in the cell cycle, if there's a mistake at the end, the cell dies. So that means that it needs to make sure that everything is organized in a nice, easy package. And so chromosomes are continuous strands of DNA that contain your genetic information. And again, they only appear, sorry, my throat is a little weird today. But they only appear when your cell is about to divide. How many chromosomes do we have? We have 46 chromosomes, okay? They have this kind of, so here's an image, right? And then here is here's a drawing, not an image. The one next to it is the image. And you can see that they all have this familiar looking X shape. We'll talk about that when we go through the steps of mitosis. But each half of the chromosome contains an identical piece of DNA. And that is because of the S stage of the cell cycle. So again, the reason why these chromosomes appear is because your cell is literally making a copy, and the X shape only is there when there is a copy of your DNA. Otherwise, there'd be no reason to have that little extra piece of the X shape. So the two identical halves are called sister chromatid. So you can see in the picture here, this half and this half. These are called sisters because they are they are copies of each other. And they are held together by this very large protein structure called the centromere right in the center. Because you don't want these chromosomes to accidentally get mixed up. You want to make sure that, if this is chromosome 1, you have a copy of chromosome 1 here. You have an identical copy of chromosome 1 there. You want to make sure. Why are they called sisters and not like, I don't know, clones or something? Because technically, and again, we're not talking about this yet, but there are reasons for it. I guess I probably shouldn't have said that. That sounds more mysterious than anything else, but we'll talk about it later on. All right. So the ends of your DNA and the chromosomes are protected by these little tiny caps, these protective tips called telomeres. Okay. And I call them junk DNA because they're kind of useless. They're just, a bunch of A's, T's, C's, and G's. And the telomeres are protecting your DNA because every time your cells divide, it's going to lose a little tiny bit of your genetic information. Just a little tiny bit. You might think, holy crap, that sounds scary. But think about it. You have a ton of important genes on your chromosomes. None of them are near the ends. They're all going to be near the middle. You don't have any important genes that are right at the very end of your chromosomes or right at the very top of your chromosomes. And there's a reason for that. Because telomeres exist at the top and bottom of your chromosomes, and they're there because every time your DNA goes through this process of manufacturing a copy of itself, it's going to get a little bit shorter and a little bit shorter. And we don't want you to all of a sudden, for some reason, to start losing important genes. So when your telomeres run out, right, like once you've gotten through your telomeres, your DNA can slowly get eaten away. There are genetic disorders involving telomeres like this. We have a feeling, and again, this is all purely speculative. People have been doing a ton of like work on telomeres. Some organisms actually add telomeres to their DNA. We do not have that enzyme to do that in our bodies, but some organisms do, so they can be perpetually like immortal almost. We don't have that. But there's an idea that aging and our cells, breaking down and getting worse at doing things might have something to do with telomeres getting used up. So, what are we going to label? So, please draw these weird little squiggly bits like this. And there are 5 main parts. 1, this would be your telomere here at the end. 2, that is your centromere right there in the center, that little red thing. 3, we call this the short arm of your chromosome because it is literally the top part of the X that's really short. 4, we call this the long arm of your chromosome. And then 5, these are sister chromatids. And so it's the entire thing. It's the short arm and the long arm of this side, and the short arm and long arm of that side. That combined, those are called sister chromatids. All right. So if you have any questions, let me know.